Hello, and welcome to the Journal Webinar Series of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. I'm Ann Edall with the Soil Science Society of America Journal. Thank you for joining us today for our webinar, Soil Biological Health is Dependent on Soil Properties and Management. Before we begin, I'd like to mention that questions can be entered into the Q&A section, and the moderator will address them at the end of the presentation. The webinar will be recorded, and the recording will be emailed to all attendees after the session. Now, I am happy to introduce our moderator for today's webinar, Christine Sprunger. Dr. Sprunger is an assistant professor of soil health at Michigan State University, based at the W.K. Kellogg Biological Station. Dr. Sprunger is also faculty within the Department of Plant, Soil, and Microbial Sciences, and is a faculty member within the Plant Resilience Institute at Michigan State University. Her research focuses on the intersection of agriculture and the environment, where she investigates how climate change impacts crop production, nutrient cycling, soil food webs, and rhizosphere dynamics. In addition, much of her work addresses how crop diversity, perenniality, and reduced tillage contributes to important ecosystem services, such as soil carbon sequestration and nitrogen use efficiency. Dr. Sprunger also conducts interdisciplinary research with soil scientists to understand farmer perceptions of soil health, adaptation, and climate.
Thank you, and thank you all for taking the time to join us today. We are excited to continue this monthly webinar series, which features the latest research published in our society journals. As Ann Edall mentioned, please enter any questions in the question and answer section, and we will address them at the end of the presentation. Without further ado, I'd like to introduce today's speakers, Dr. Manandir Singh and Benjamin Ajay. Dr. Singh is an assistant professor and cropping systems agronomist at Michigan State University. Dr. Singh's research, extension, and teaching program focuses on current and emerging issues in cropping systems of Michigan, with an overall goal to improve the productivity, profitability, and resiliency of these systems. Current research is focused on management practices such as crop diversity, varietal canopy, planting methods, planting time and density, harvest decisions, and plant pathogen interactions in corn, soy, and wheat-based cropping systems. Benjamin Ajay is a doctoral candidate in Cropping Systems Agronomy Lab at Michigan State University. Mr. Ajay's research is focused on building resilient cropping systems through the integration of sustainable management practices to enhance yield and profit. His research evaluates strategies including cover crops, nutrient management, soil amendment, tillage, and crop diversity. Today, Dr. Singh and Mr. Ajay will discuss how tillage influences soil biology and overall soil health. In particular, they will address the question, does higher crop diversification influence soil microbial composition and abundance? They examined soil biological health properties and field history data from 310 grower managed fields in Michigan to help answer these questions. The report was published in the March-April 2024 issue of the Soil Science Society of America Journal. Dr. Singh and Mr. Ajay, welcome, and the floor is yours.
Thanks, Christine. Thanks for the nice introduction. And as you said, welcome everyone. It's our pleasure to be talking to you all on this topic today. It's recently published, as Christine mentioned. So our goal is to go through some of the slides here quickly, and then really entertain questions, that you all have on this topic. And I'll do the first intro here, and then let Benjamin talk about this work that he's been doing as part of his PhD program. And as all you are aware, soil health concept is it's still an emerging concept, but there's a lot of interest, and that interest has grown exponentially over the last decade or so, right? Both in the public as well as in the private sector, and not only in the US but across the globe, right? And the basic concept, as you can see on this slide, is the definition, so to speak, is pretty much, soil health is a continued capacity of the soils to function as a vital living ecosystem, right? And that sustains multiple forms of life, right? For plants, for animals, as well as for humans. So a very broad concept. And for me, I think, what are our end goals? And to me, the biggest one is crop productivity, right? How we can benefit from this approach to improve our agronomic output, right? And productivity is right there. Profitability is probably in that same domain. Resiliency of our systems is right there. And then there are other ecosystem services, right? Enhanced biodiversity, improved water quality by minimizing nutrient losses, and mitigation of greenhouse gas emissions. I think all of those concepts sort of come together. So it's when we think about components, it's a very complex system, right? As you can see on this figure here, when we typically think about soils, we are pretty honed on the chemical portion, right? When we as farmers, send out our samples in US, typically we get the nutrient analysis, including pH and available nutrients. Every now and then we look into the physical properties, right? Soil structure, water holding capacity, porosity, things like that. But soil biological component often is being neglected, right? And that's it's a very important component of this whole system. And the critical aspect here is that it is very sensitive to management. And there is a lot of data that is out there coming from research plots, research stations, but there is lack of data from grower managed fields. So that's what we out to do in this project. And I will let Benjamin sort of explain what we did, how we did it, and what we found out. Over to you, Benjamin.
All right. Thank you, Manny. Welcome everyone to the webinar again. So I would like to begin by giving a little bit of background to approaches of studying soil biological health. So like we mentioned, it's an important component or aspect of soil health that has not been, studied to a much deeper depth, especially looking at data from grower managed fields. And so traditionally we have two approaches to studying soil health. The first approach mostly involved the measurement of indicators that reflect biological activities. And you have so many indicators. Example of those ones include proteins, enzymes, and for example, POXI. And so many different kinds of studies have been conducted to relate some of these indicators into functions. For example, POXI have been shown to have a linear correlation with soybean yield. And so if you're able to improve your POXI levels, it's able to significantly improve soybean yields. The second approach to studying soil biological health involved the direct measurement of microbial populations. And it's important that we're able to quantify how much biodiversity we have within our soil microbiome. That way we're able to modify our management strategies so that we don't harm our soil biological health. So within this domain, we have several techniques or several methods to estimate direct amount of soil microbial populations. And one of the most commonly used approaches is what we call the PLFA or the phospholipid fatty acid method. So this method is popular because it has some benefit as compared to other ones. For example, it has a short turnover times and able to provide a snapshot of our soil microbiome in terms of the actual numbers within a short time period. Additionally, this method is also sensitive to changes in soil properties. For example, as you move from across different soil textural classes, this PLFA is able to detect how your soil microbiome is changing. Also, it gives us this quantitative output, so you're able to quantify the actual numbers that we have within our soil microbiome. For example, it will give us an idea of how much AM fungi are shown over here that we have in the soil, as well as how much soil fungi we have in the soil. And these microbes perform several functions within the soil. Example, nutrient regulation, pest and disease suppression, and so many other ones. It also gives us an idea of how much actinobacteria and also fungi that we have, eukaryotes that we have. Eukaryotes are the bigger multicellular organisms in the soil, such as nematodes and earthworms. And so like many mentioned in the previous slide, soil biology or soil biological health is hugely or significantly impacted by management. And so I would like to mention a little bit about how management affects soil biology. So your management can either enhance or reduce your soil biological health. And how does that happen? So you can either degrade or enhance your microbial habitat. So as a background, microbes mostly lives in soil aggregate. And so practices that will reduce your soil aggregate structure will harm your soil biology because it will disrupt the microbial habitat. And practices that will cause more traffic on the soil will cause more compaction, and that have effect on the porosity as well as the moisture dynamics within your soils. And so it's important to understand some of these dynamics to be able to identify which practices will enhance your microbial habitat and then which practices will not do that. Another way that management influences your soil biology is to either add, remove, or manipulate food resources. When we talk about the food resources, we are looking at organic matter, we are looking at residues. And so management strategies that leaves a lot of residues and organic matter on the soil surface provide a lot of biomass for soil microbes. And so if you remove residues from the soil, you are taking away food resources that the microbes can use. If you continuously till your soil, you are accelerating the rapid breakdown of your soil biomass, and then the food resources become less available, or like within a short space of time, the food resource for the microbes runs out. As compared to like other practices such as cover cropping and residue retention, if you put more cover crop in your diversity or within your rotation, you're able to provide a lot of residues, a lot of organic matter for soil microbes, and that enhances your microbial, your soil biological health, and the overall soil health of your fields. It is important to mention that the management impact varies based on your environment. And environment, we are looking at what's your soil properties like, what is the soil texture, what is the soil drainage, what is the porosity, and all those kind of things. And so it's becoming increasingly important that we examine how some key management practices affect our soil biological health and overall soil health, and most importantly at the farm scale, where we are using data from grower managed fields as compared to experimental studies or experimental research fields. And so the objective of this study was to understand which soil properties and also which management practices drives our soil biological health and the overall soil health of our soils. Specifically, we wanted to understand how soil texture affects microbial populations. We also wanted to understand how tillage affects microbial numbers. And lastly, we wanted to examine how crop diversity affects our microbial numbers. So how did we conduct the study? So the study began in 2019 in Michigan. We sampled more than 300 grower fields, as Christine mentioned before, and the samples came from 29 counties, and the shaded areas on the Michigan map shows the counties where we pick our soil samples from. Sampling began in fall 2019 through to spring 2021. Details of the number of fields within each sampling time can be found in the paper. So our target soil sampling depth was 0 to 15, and we took samples from representative areas within the field. You can imagine some of these fields are huge, but so in the sampling process, we avoided problem areas, but then make sure the soil sample come from areas within the field that capture the overall variability within the field. And at each soil sampling time, we took the GPS coordinate of the field that we sampled, and the GPS coordinate was used to extract additional data for the analysis that we performed. So the soil sampling followed the soil health sampling protocol, and all samples were shipped to University of Missouri Soil Health Lab, and that's where we did the soil analysis that we did. So this study is focused heavily on the component of soil biology, which involved the direct measurement of the microbial numbers. And so the initial analysis, the first analysis was PLFA, so knowing all the different broad categories of microbes that we have in the soil. Additionally, we also did we measured other indicators that reflect biological activities, which are a component of soil biological health. And our focus over there was on POXI, we also did PMNN, we did organic carbon, and we also did water stable aggregation. Details of the extraction protocols of all this analysis can be found also in the paper. So lastly, we also did we got the soil texture data from the soil web survey. So we took the coordinates that we had from the field that we sampled, and then we put those coordinates into the soil web survey, and we extracted the percent sand, the percent silt, and the percent clay, and then we were able to identify the soil texture for each field from that information. So our broader goal was to understand how agronomic management affects the indicators that we are measuring. How does management impact your soil health and soil biological health? And so we sent our field history survey sheet to growers, and we had an extensive or a list of management strategies or management information that we wanted to obtain from the growers. Example of the survey sheet that we sent to the growers is what is shown over here. So we wanted to understand their crop history, what's their crop rotations like. We also wanted to get information on their cover crop strategy. Do they include cover crops in their rotations or not? We're also interested in tillage. We wanted to understand how their tillage operations. We were interested in manure. We had questions on residue management. We had questions on fertilization and so many other ones. But I mean, as part of the difficulties in this kind of research, we did not get response from a lot of all the management strategy that we sought to identify to obtain. And so only few growers responded to the cover crop and the manure application, as well as the other ones. And so we ended up having enough information or data on tillage and then crop rotation. So that became the focus of the study. So we have at the end we had like three main things to look at as if it reflects in the title of this presentation, in the title of the paper. We had our focus was on texture, on tillage, and crop diversity. And for texture, we had three classes of texture. We had fine, medium, and coarse texture soils. And this classification was based on the soil texture class that each of the fields were. And so from this soil texture triangle over here, for fine, medium, for fine soil, these are represented by the red stars over here. So if the texture as identified based on the percent sand, silt, and clay was, if the texture was clay, then that soil was classified as a fine soil. Again, if it was sandy clay, clay loam, silty clay, and then silty clay loam, those soils were classified as fine soils. Soils with texture classified as medium are represented by the green stars over here. So for example, if your soil is sandy clay, loam, or silt loam, or silt, those soils were classified as medium. And finally, the yellow stars represent the coarse textured soils. So soils such as sand, loamy sand, and sandy loam were classified as coarse textured soils. So the next thing was tillage. So we had two levels of tillage. We have tilled and then no till. So for every field that had experienced at least 1 year of a tillage pass, we classify those fields as tilled. And then for fields that had not received any tillage pass within the 3 years that we examined. So we took data from 2016 to 2018. That's the management information. So the idea was to understand how this management has affected or impacted the soil biology before our sampling time. And so within the 3 years, if there are no tillage passes, we classify those soils as no-till. And finally, we had the crop diversity index. So crop diversity index estimate the number of species within the 3 years that we examined. So from 2016 to 2018, if the same crop was planted, we gave it a diversity index of 1. So if you have like continuous corn from 2016 to 2018, or we gave a diversity index of 1. If we had like 2 crops being planted between 2016 and 2018, we gave a diversity index of 2. So a soybean corn rotation will have a diversity index of 2. And lastly, if the rotation had at least 3 crops, we gave a diversity index of 3. So typically those rotations were corn, soybean, wheat predominantly, but other times we have some other crops coming within the mix. But it's also important to mention that this diversity index does not include cover crops. So these are mainly the cash crop that we examined because we had limited information or limited data on the cover crop practices. And so we analyzed the data by pulling the data across the years that we took the soil samples. So data was put across all the years and across all the sampling times. So what did we find out? So first here, I'm showing the effect of texture on microbial populations, and I'm representing those with split plots. So the solid line in the middle represents the median, and the diamond on top represent the average for each indicator. On the top panel, I have the total PLFA, which represents the total microbial abundance within the soil, and I have the other broad categories for fungi and bacteria on the top panel, and on the lower panel, I have A and fungi, I have actinomycetes, and I have eukaryotes. And these 3 different bars represents the different soil texture classes. So you have coarse texture soils, you have medium, and then we have fine soils. So overall, we found like minimal impact of texture on most of the soil microbes. As you can see, overall microbial abundance was similar across the texture classes. But if you look at the individual species, most of them were similar across all texture classes, except for actinomycetes, which was significantly higher with fine textured soils. And you know, fine textured soils has higher amount of clay, and clay is able to hold a lot of nutrients to their surfaces and also able to hold more organic carbon. And so organic carbon is a substrate, and the nutrients are something that microbes need. And so that could be part of the reasons why we found higher amount of actinomycetes with the fine texture soils. So we also look at the effect of texture on the other soil health indicators that reflect biological activities. So we have organic carbon, we have POXI, and we have PMN. So again, we found minimal impact of texture on these biological indicators. However, as you can see, there's an apparent trend of increasing numbers of these indicators. You can see the average as we move from coarse texture soils, it's increased slightly with some of these indicators. Now, the next thing that we look at is the effect of microbial populations on the effect of tillage on microbial population. And this was the dominant thing that we found in our study. We found that tillage significantly reduced soil microbial numbers. So overall, the total microbial population, the total microbial abundance, as shown in this top figure, but total microbial abundance was about 17% higher in the no-till fields as compared to the till fields. The same applies to all the other indicators. Bacteria, fungi, fungi, actinomycetes, and eukaryote populations were significantly low under till as compared to the no-till fields. Another interesting thing was the indicator that reflects biological activity. As you can see over here, organic carbon, POXI, and water stable aggregation were significantly low in the till as compared to the no-till. And this goes back to the slide that I mentioned before, the effect of management on the role that management plays in biological health. So continuous tillage disrupt your aggregate stability, and so you disrupt the microhabitat. It's also enhance the rate of decompositions, and so it significantly affects your soil microbiome. As you can see, so organic carbon was low, POXI was low, and WSA. So for water stable aggregation, no-till was more than about 20% more stable as compared to your till fields. And like I mentioned, aggregates are what holds or where microbes mostly reside. And so you don't want to disrupt your aggregate stability too much. So now we look at the effect of crop diversity on microbial populations. And the top panel, we have the total microbial abundance. I have bacteria and I have fungi. And at the lower panel, I have fungi, actinomycetes, and I have eukaryotes. And you can see that lower crop diversity affected some soil microbial compositions or microbial groups. For example, we found lower amount of fungi and also eukaryotes with low crop diversity. You could see as you increase your crop diversity, fungi numbers increases, and the numbers of eukaryotes also increases as you increase your crop diversity. And increasing crop diversity means you are having multiple source of food resources in code for the microbes, as opposed to having just the same crop over a period of time. And so if you have like a corn soybean within the rotation, different microbial numbers, different microbes come into the mix, because different microbes will attack these residues or these organic materials differently. And so if you have the same kind of residue or the same kind of biomass over a long period of time, it doesn't enhance microbial diversity in the soils. And if you look at the other indicators that reflect biological activity, we found a similar trend. We found that POXI was significantly lower with less crop diversity. And POXI is part of the active pool of carbon where microbes are actively working on them. And so if you have less crop diversity, you don't introduce different mixes where they have different chemical compositions where different microbes can attack. And so and then POXI is also important. This information is super important because, like I mentioned before, some studies have found that higher levels of poxy correlate to higher soybean yield. And so if you're able to introduce some of these, introduce more species diversity into your rotations, you're able to build more poxy and you'll get some benefits such as crop yield. Again, we also found an increasing trend of higher indicators with higher crop diversity. As you can see, the organic carbon content was increases as we increase our crop diversity, and also PMN also increases as we increase our crop diversity. So before we conclude, I would like to mention some limitations of our data set. So we look at broad categorization of tillage as no-till or tilled. We did not have the actual data on the number of passes and the frequency, and so we did not examine like tillage intensity as part of our data set. We also did not get a lot of response from growers in terms of manure applications, and so we did not examine manure amendment as part of our data set. And manure amendment can significantly affect your soil biology as well. We also did not get information on residue management as well as cover crops, as they were we did not get enough data from the growers. But residue management, as I mentioned before, and cover crops are some of the strategies that either enhance your biomass availability, which are your food resource for the microbes. And so some of these things can also significantly affect your soil biological health. And it's important to mention that cropping system in Michigan and several other states can be very diverse, especially if you include cover crops in their rotations. But our limitations over here, we only have like 3 levels of diversity, 1, 2, 3. But then some species diversity can be more higher, can be up to like 5, 6, 7, and all those ones can affect your soil biology. So in conclusions, we found that tillage was a dominant driver of soil microbial community. In this data set, it significantly reduced all microbial numbers. It reduced total microbial numbers and also reduced total the different species of microbes. It also significantly reduced some other indicators of soil biology, such as organic carbon, poxy, and also the stable aggregation. We found that crop diversity and soil texture had minimal impact, but crop diversity especially impacted a key indicator, which is poxy, and that is important to highlight that given the benefit of poxy or the role poxy plays in crop and yield enhancement, it's important that we highlight that. So to provide some recommendations, given that tillage was a dominant driver, our first recommendation is to minimize disturbance. And how do we minimize disturbance? You can minimize disturbance by reducing tillage frequency. So if you do fall and spring tillage, you might want to cut down by doing just spring tillage and not doing any tillage in the fall after your harvest. You can also reduce the number of tillage passes. And another thing that we can also do is to progressively switch from a dominant conventional tillage to a no-till conditions. So to enhance crop diversity, we want to maximize biodiversity. And how do we do that? You can increase your crop diversity by having more cash crops in the mix, moving from continuous corn or continuous soybean to a corn soybean rotation or corn soybean wheat rotations. Another way to maximize improve your crop diversity is to plant more cover crops. You can do single species cover crop like planting cereal rye after corn, or doing more cover crops mixes. That would also be useful. And you can also integrate livestock within the mix. And all these recommendations will help you to achieve the 4 soil health principles, which are to maximize continuous living roots, to maximize biodiversity, minimize disturbance, and also maximize soil cover. So I will pass it on to Manny for him to wrap up here for us today. Thank you.
Yeah, so yeah, very quickly, again, this was a lot of work, so we would like to acknowledge Michigan Department of Ag and Rural Development, who provided funding for this work. A lot of MSU extension educators helped with collection of these samples, so we really want to acknowledge them. It is a lot of work to do this sort of work at farmer fields. As mentioned before, the article is published in the journal, so please check that out. We also have a bulletin, extension bulletin that is available on our website, agronomy.msu.edu. And with that, we'll take any questions.
Great, thank you so much for that wonderful presentation. At this point, we do have some questions that we've received in the chat. We'll probably take about 5 to 10 minutes to answer those questions. But for those of you that need to leave early, the full recording will be available after we close the webinar. So the first question is, did any of your study fields use biostimulant products?
So biostimulants. So yeah, we did not get, or I would say we did not go look for that information, but my thinking is that, yeah, most of these fields would not have received a biostimulant application. This is back like 5 years back or so, 2016, 17, 18. And we have growers who are using them, but I would say this probably would be a minority of our data set, I would think. And that's a topic we are actively looking into, what are the implications of applying biostimulants short-term as well as long-term in our cropping systems, right? So we have a lot of ongoing research on soybeans, on corn, or even on small grains, looking at the impact not only on crop productivity, but on soil health consequences as well. And I think Christine and I have some projects dealing with that. So we'll have some more data, I think, on the impacts of those biostimulants here down the road.
Yeah, that's, I think, a really important topic that a lot of people have questions about. And I would say that the science still isn't quite there to fully be able to address that, but it's definitely something, as Dr. Singh said, that we are working on. So another question is, can you help me understand how you estimate potentially mineralizable nitrogen? And then also, is there a way that you could determine how cover crop biomass, for example, might influence potentially mineralizable nitrogen?
Benjamin, you want to take that?
Yeah, so the first part of the question is how to estimate POXY. And
oh, it's not POXY, it's potentially mineralizable nitrogen, PMN.
Okay, so it's a good question, but I don't know the protocol offhand as to I don't know whether the question is the extraction process that they want to understand or the importance of it. But the extraction protocol, I don't have it offhand. I mean, it will be fine. I think I explained it in the details in the paper, how the process is extracted, how PMN is, analyzed in the lab. I don't know if Christine have more insight on.
Yeah, I can say a little bit more. So the potentially mineralizable nitrogen protocol is really an anaerobic assay, and you can do this over like a 1 day kind of incubation, a 3 day, or up to 7 days. And that will get you a sense of more recently released nitrogen into the system. But I think your question around the impacts of cover crop biomass on potentially mineralizable nitrogen is a really important one. And it's probably a question that will actually take multiple kind of protocols and approaches to really address. For example, there was a great paper, Sivers and Cook 2018, that was also published in the Soil Science Society of America journal. And what they did was they used root simulator probes that actually are able to detect plant available nitrogen. And you can do that very close after cover crop termination, for example. So if you're interested in really understanding more of those methodologies, definitely check out that paper. We can share that information in the chat as well. Okay, so another question is, did you notice any trends in fields that use certain herbicides or insecticides on microbe populations if you received pesticide history?
Yeah, that's a good question. I'll let Benjamin maybe explain later, but I feel like we did not get enough of that information to make any sort of conclusion based on our data. I assume most of our growers do apply pesticides, for sure, herbicides. Some do apply fungicide as well as insecticides. I don't think our data set is able to answer that question. Is that right, Benjamin?
Yeah, that's right. We don't have the management data on pesticides and herbicide applications in. It's part
Yeah,
of
I
the.
know it's an active topic of research. I don't know, Christine, if you have seen any data on that. I know some of my colleagues around here are actively looking into these implications.
Yeah, we see a little bit where, I study beneficial nematode communities, and there's definitely an effect on different kind of insecticides and nematicides are targeting the like root knot nematodes, for example, but they do kind of sometimes have adverse effects on beneficial nematodes. But I would say, again, we need more consistent long-term trials to really get at that question.
Yeah.
Okay, another question is, which organisms are included in the eukaryotes category that are different from fungi? And did you measure those in this study?
So, I mean, broadly, the eukaryotes include like nematodes, earthworms, but then the PLF analysis did not isolate the individual nematodes or earthworm composition within the broad categorization of eukaryotes. But yeah, those are some of the organisms within the eukaryotes domain.
Great. Okay, another question is, what was the impact of tillage on the organic matter decomposition through microbial activity?
If I understand it, what's the effect of?
The impact of tillage on the organic matter decomposition through microbial activity. So maybe the best way to answer this is thinking about the impact that tillage had on like soil respiration, for example.
Right. So tillage accelerates the rate of respiration, accelerates the rate of biomass decomposition by microbes, because first of all, the tillage breaks down. I mean, that's like more physical, but then it breaks it down more into like more smaller particles. Then it increases the surface area for microbial attack and microbial actions. And so I think that will be a way that I will go about it. Like, so tillage will first of all break residues or organic matter into smaller particles first. That's more physical. And then microbes will then attack, releasing more enzymes that are like work on these smaller surface, these high surface areas, and then accelerate the rate at which they decompose or break them down.
Yeah, and I think I would like to like it's a broader question, right? Like implications of tillage. We are showing again differences, but what we really wanted to do was like look into the intensity piece, right? Because there's a lot of middle ground going from a full conventional to a no-till system. And as Benjamin mentioned, we didn't have, I think, as much data in that domain, to really look into, where to cut. But again, I believe it starts with, thinking about, frequency and then number of passes within your tillage operation in the fall versus in springtime, right? All of those will have more implications. So again, we need to dig a little deeper into some of these implications, what we are doing with tillage on some of these parameters, right? But again, a good starting point for us to dive further in.
Great. So here's another follow-up question, and this will actually be our last. I'm sure that if we didn't get to your question, that Manny and Benjamin would welcome any emails, because I see a lot of really good and detailed questions here. So please reach out to our presenters, and I'm sure they'd be happy to respond to your questions. But one last question: what management practices would you like to follow up with any additional trials to look at management impacts on soil biology?
Yeah, that's a good one. There's probably a lot, and maybe I'll let Benjamin, I think, give his thoughts. But I think we were just talking about tillage. That to me is a big one, and even crop diversity piece. Benjamin alluded to some of these challenges, going to more diverse systems, and we have plenty of those here in Michigan. So right off the bat, I think I can think of that. And Benjamin, I'll let you explain more.
Yeah, definitely. I mean, doing more on tillage, doing more on diversity, including more cover crops in the mixes, even how to integrate livestock and all those kind of things. It needs more in-depth research. Also looking at some other stuff like manure amendment and residue retention or residue management. These are things that we can do more research and understand and elicit the exact effect or influence on some of these biological indicators that we are talking about.
Yeah, and I think one thing I would say here in northern systems, in Michigan, winter wheat is probably our best cover crop, because you can take it to cash, and it's a cash crop, and it provides pretty much all the other ecosystem services, productivity benefits, and probably it's tied to the soil health, so implications of, again, residue management, leaving it in the field versus harvesting, only grain versus straw yield. We have growers who do take straw from their winter wheat as well. So those implications, on the soil health and relating that to productivity. And again, I think that's another missing piece here. Benjamin did allude to that, but yeah, how does the soil health parameters relate to productivity? That as an agronomist is my end goal. How we can help our farmers improve their productivity while being profitable, meaning that they can cut down on input costs, right? And these all are, I think, sort of connected things that we continue to look into.
Great. Well, thank you again to our presenters, and thank you all for joining us for today's webinar. As a reminder, the recording will be made available and sent to all attendees soon. Thank you again, and have a great day. Take care.
Thank you.
Thanks, everyone. Have a good day.